differences between plant and animal cells. Plant cells have chloroplasts, central vacuoles, and cell walls. Animal cells do not. Centrioles have animal cells, and flagella. Okay, any other questions?
The difference between plant and animal cells, I mentioned how the plant cells look like they're brick walls, and animal cells are kind of
Amorphous, yeah. Yeah, more, you need something for a cell wall, to make those bricks. Yeah. I'll look at it. But anyways, okay, I left off talking about the membrane. Diffusion is, again, with the gradient, passive transport, always, diffusion of a liquid in a liquid, we've seen that. Diffusion of a gas in a gas, we've seen that. Active transport, you need energy. Energy is required. Active transport goes against the gradient. Okay, that's very important. And then bulk transport. Bulk transport referring to moving mass quantities of metabolic waste products and or nutrients into and out of the cell. Mass quantities of that. Had bulk transport.
Can you say that one more time? I'm sorry.
Bulk transport.
Yeah.
Mass quantities of material exiting and or entering the cell. If it's exiting the cell, it would be known as exocytosis. If it's entering the cell, it would be referred to as endocytosis. And again, exocytosis, mass quantities of metabolic waste products exiting the cell. Endocytosis, mass quantities of nutrients entering the cell. Diffusion and osmosis. And, you can have diffusion without osmosis, but you cannot ever have osmosis without diffusion. Follow that? I'll say that again. Diffusion, you can have diffusion without osmosis. You can have diffusion.
I know 3 times, but I just
Right, yeah.
Thank you.
But you cannot have osmosis without diffusion. Simply stated, osmosis needs a membrane. Okay? And again, I can't emphasize this enough, biologically, that membrane is very, very important, okay, to establish gradients. Okay. And water. Osmosis deals with water and a membrane. Diffusion. We saw diffusion in a liquid and a liquid. malachite green, those drops of malachite green added in water, right? High to low concentration. Diffusion of a gas in the gas, the coffee aroma. There was no membrane separating but osmosis, you need a membrane and water. The osmotic pressure, you need some type of pressure being exerted. In biological terms, very, very important for that membrane to establish those gradients. Osmotic pressure, those were the things that hyper or isotonic. Iso refers to equal, so equal pressure on either side of the membrane. And may have you do similar, if I had I'll do this again. 2% sucrose in a cell, 98% water. Put this in 3 different beakers. This would be isotonic, equal 2% sucrose, 98% water. In the solution, just as much water will enter the cell than will exit the cell. Again, just the same amount of water will enter the cell than will exit the cell. Isotonic, iso equal, 100% water. 100% water. Most of the water will enter the cell. You are going to have some exiting the cell, but the net movement is entering the cell. This is hypo. Hypo. Hypo refers to lower concentration of the solutes in the solution. It's all in reference to the solution, not the cell. Hypotonic. And then finally, 90% water and 10% sucrose. Most of the water will leave the cell. You are going to have some water entering the cell, but the majority of water will exit the cell. This is known as a hypertonic. Hyper, higher than or greater than. Again, this is all in reference to the solution. a hypertonic solution has higher concentration of solutes. What's a solute?
Diluted solution.
Right, exactly. Anything that is dissolved in a solution, and that's why they're called solutes. Again, I can't emphasize enough. It's all in reference to what's going on in the solution itself. Hypotonic solution. Animal cells. Animal cells prefer being bathed in isotonic solution. The plasma of our blood, the red blood cells in the plasma of our blood prefer equal solutes. So most animal cells prefer equal solutes. Most plant cells prefer hypotonic, somewhat swelling up. Why? Right, good. Turgor pressure. To establish that turgor pressure. And that's what happens whenever we forget to water our plants. They lose that turgor pressure and they start to rot. So on the lab practical, I'll probably have one station, actually a couple of stations pertaining to turgor pressure. The loss of turgor pressure or the wilting of a plant equates to, and you would say, the loss of a turgor pressure. For the lab practical, there's going to be 25 stations around the moon in the lab. You go from station to station and answer the question. For this first lab practical, know the parts of the microscope. Have one station with a microscope and label on the ocular lens. And the question may be, identify the labeled part of the microscope. And you would say ocular lens. I would accept eyepiece. Be able to compute the compound microscope as it's set. I'll have the ocular lens pointed to the, so you can see 10 times. And then if I have the 4 times objective locked in, you would multiply 10 times the 4, so 40 times. The revolving nose piece, the iris diaphragm, the fine focus, the coarse adjustment, the stage, mechanical stage. Don't worry about the condenser. I don't want. We have 6 cell models. 3 plant cells and 3 animal cells. So expect testing or be able to identify at least 6 of the organelles. The mitochondria, Golgi body, endoplasmic reticulum, nucleus, nucleolus. The cell wall of a plant cell. The plasmodesmata, and they have a wall-to-wall junction, and that would, with a label on the plasmodesmata, and if you identify that this wall-to-wall junction, so only plant cells will have gap junctions known as plasmodesmata. And 3 or 4 stations on the reagents that we use. I'll have a series of potassium iodine bottles, Biuret solution, Sudan number 4, and Benedict solution. And I'll ask you identify which reagent will be signified for starch, or indicator for reduced sugar. Question? Yeah, the bacteria. The planktonic organisms that I think we're all set here. Plankton. If I ask you, name one of the 2 main types of planktonic organisms. That would be phyto, phyto referring to plants, and zooplankton. One of 2. Zoo referring to animals. Turgor pressure, I mentioned as well. If I have a wilted plant, what's counteracting this wilting of a plant? And or if I have a mock cell, and give you these parameters or these values. If this cell has 98% water and 2% sucrose, and it was bathed in a 90% water solution with 10% sucrose, what type of tonicity would you expect to see? And you would say hypotonic or hypertonic. Hypertonic. And the same thing for diffusion of a liquid in a liquid. We find more or less everything in those labs, all those labs detect is fed. So, but anyways, plant cells tend to prefer hypotonic solutions because of the plasma desmata, or the plasma membrane. It wants to be almost adhered to the cell wall in a hypertonic solution. Right, we saw that. Plasmolysis occurred when you add a saline solution to the healthy LAD plants. And I have this description or this cartoon of the animal cell versus the plant cell. In hypotonic solutions, that's normal for a plant cell. These animal cells will be lysis. Lysis. Whenever you see the word lysis, you should be thinking to split apart. Normal for isotonic in a plant animal cell, and shriveled up in a animal cell. If it continues to shrivel up, it will become what is known as cremated cell, and or in a hypotonic solution, it's referred to as a lysed cell. And this is in a hypertonic solution. If the shriveling of a animal cell, and then it adds more of a isotonic, it will come back. But if the animal cell gets cremated, that's the death of the cell. Just like you saw certain with the plasmolysis. It will swell and right. If it lyosis, it splits apart. Once that membrane is split apart, that's the death of the cell. If that membrane is able to rebound, right, and keep its structural soundness, it will return back to normal. Turgor pressure, again, hypotonic situation. That's the pressure of the cell wall. I'm sorry. The pressure of plasma. Membrane. Plasma membrane. against the cell wall. And again, this is hypotonic solution. With the saline solution, this would be, these, I showed you these before, and you had, I saw a lot of really nice cells. This cell will come back. This will not. I can definitely see the plasma membrane has ripped apart. This cell here apparently has the plasma membrane intact. So if you add more water, much more, flood it with water, or even distilled water, this will, the turgor pressure will come back and almost adhere to the cell wall again. These cells most likely will not, and definitely this cell will not. If you see the plasma membrane ripped apart, chances are that that's the death of that cell. But again, if you see the plasma membrane intact, if you add more water, good chance for it to return back to a healthy cell. Okay, to structure, fibrous and primary and secondary structures of proteins or fibrous proteins, tertiary and quaternary structures of proteins or the globular proteins. They're all proteins. Again, in on Wednesday's exam, I may have a short answer, labeling or drawing a plasma membrane, and incorporate all of the structural proteins embedded in that plasma membrane. But again, what I'm be looking for is the hydrophilic heads and the two fatty acid tails, which would be hydrophobic. And then the arrangement, head to tail and tail to head. And then the embedded with the molecular gates, which would be the transfer of proteins. Facilitator diffusion, no energy is required. This is a passive transport. Concentration gradient. Again, active transport. The best you'll learn in anatomy and physiology, the resetting of any neuron. You need the potassium sodium pump to reestablish those gradients within that neuron. Once the depolarization, repolarization occurs, and to reset that neuron, you need against the gradient. So ATP is needed, and energy is needed. Again, against the gradient, you need energy in form of ATP. And again, this would be embedded in a plasma membrane. Whenever you see these circles and tails and circles, phospholipids membrane. In exocytosis and endocytosis, vesicle, membrane-bound vacuole, okay? It would be analogous to a vesicle would be a membrane-bound vacuole, okay? Mediated bulk transport, I refer to it as moving mass quantities of material into and out of the cell. Endocytosis is a pinching off. Think of these as advantageous ions or nutrients. Okay? A pinching off. This would represent the plasma membrane. Again, bilipid phospholipids in an arrangement. Pinching off of the plasma membrane. And then again, once that vesicle is inside the cell, okay, the plasma membrane, all these phospholipids will disengage and again be incorporated in the plasma membrane again. And the Golgi body produce these vesicles for exocytosis. So exocytosis, you would think that all of these metabolic waste products, again in mass quantities, or exiting the cell. And that little video I showed last time showed portion of exocytosis. But those would be metabolic waste products. Carbon dioxide would be dealt with in mass quantities, leaving the cell. So, and then picked up by a capillary and become incorporated and being in a vein pumped out